Hey guys, welcome back to another session on diabetes mellitus. I am Dr. Ravi. In this session, we will learn about physiology of insulin. The endocrine pancreas is made up of roughly 3 million islets scattered across the exocrine gland, weighing 1 gram, which constitutes under 3% of the total pancreatic volume. Islet of Langerhans is populated with a variety of cells like alpha, beta, gamma, delta and E cells. Beta cells are abundantly present in islets, composing 60% of the islets, intermingled with the other cells. Higher proportion of beta cells are present in smaller islets when compared to the larger one. This means a high concentration of insulin is present in the smaller islets, and the smaller islets are located in the vicinity of blood vessel. So the spatial arrangement is also important for the islet function. Heterogeneity of the cells and the spatial arrangement of the cells allows the pancreas to exhibit the paracrine effect. So what is paracrine effect? Secretion of one hormone will inhibit the secretion of hormone from the adjacent cell. For example, insulin release will inhibit the release of glucagon, amylin release will inhibit the release of insulin, somatostatin release will inhibit the secretion of insulin and glucagon. So the islet regeneration occurs not only by the neogenesis of beta cells and ductal replication, but also by transdifferentiation of alpha and delta cells. So a novel concept is emerging that the function of the islets not only depends upon the mass and quality of beta cells, but also on the architecture and structural integrity of islets as an organ. Alpha cell secretes glucagon, and beta cell secretes insulin, amylin, C-peptide, and minute portion of proinsulin. And then delta cell secretes somatostatin, and E cells secretes ghrelin, and all gamma cells secretes pancreatic polypeptide. Then coming to the structure of the insulin. Insulin is a polypeptide hormone consisting of 51 amino acids with two chains. One is A chain, another one is B chain. So A chain consists of 21 amino acids and B chain consists of 30 amino acids, and both A and B chains are linked with the disulfide bond. So this is the structure of insulin. Then coming to the synthesis of insulin. So the genetic code for human insulin is embodied in short arm chromosome number 11 on sequence 15. So suppose this is a beta cell of the pancreas, which consists of nucleus and cytoplasm. So this is nucleus and this is a cytoplasm. In the nucleus we have a genetic material. Called DNA. So DNA, due to the transcription, mRNA is formed. So mRNA is formed, which contains the codons for the amino acids which forms the insulin. So this mRNA is sent to the cytoplasm. In the cytoplasm, mRNA binds with the ribosome. And with the help of tRNA, what happens? The amino acid sequence will form here. So this amino acid sequence, initially formed amino acid sequence, is called as signal peptide. Otherwise, it is called as signal sequence. So, what is the significance of signal peptide? So, we have endoplasmic reticulum in cytoplasm. So, signal sequence or peptide will pierce the membrane of endoplasmic reticulum. So it pierces the membrane of endoplasmic reticulum, and in the endoplasmic reticulum a single polypeptide chain is formed and it is called as preproinsulin. So preproinsulin is formed. It is a single polypeptide chain with which consists A chain, B chain, A chain, C peptide and B chain and signal sequence. So this preproinsulin is cleaved and the signal sequence is removed. Okay. So due to the different polarities of the amino acids, it tends to coil. So what happens? So it will be coiling. So this is the signal sequence, and this is beta chain, and this is connecting peptide or C peptide, and then this one is A chain. Now there is a cleavage between the signal sequence and remaining portion. So what is left? We are left with A chain, C peptide and B chain. So this is called as proinsulin. Proinsulin, which consists 86 amino acids. Now this proinsulin is taken up by the Golgi operators in the cytoplasm. So we have this is A chain, then this is the B chain, and this one is connecting peptide or C peptide. So what happens in the Golgi operators? So this is the proinsulin. So this proinsulin is cleaved. So endopeptidase enzyme will help, and the process is called as proteolysis. So the C peptide is removed from the A terminal and the B terminal. So what is left? We are left with the A chain and B chain. Here the disulfide bond is formed between A and B chain. So we are left with the matured insulin and the connecting peptide. So both this matured insulin and connecting peptide along with a minute portion of the proinsulin will bud off from the Golgi operators, and it is stored into the secretory vesicle. This is called as secretory vesicle. Okay, and it is released when it is needed. Insulin and C peptide are released in the equimolar portion. We assess the C peptide to know the plasma insulin concentration. Why we are assessing the C peptide rather assessing the insulin? Because insulin is released into the portal circulation and taken up by the liver. So before reaching into the systemic circulation, 50 to 60% of the insulin is cleared by the liver. So insulin undergoes the first-pass metabolism. So this implicates whatever the insulin is secreted into the portal circulation is not same with the concentration of insulin which is in the systemic circulation. That's why we assess the C peptide. So assessment of the C peptide helps us to discriminate the endogenous insulin from the exogenous insulin. So I had discussed in detail about the release of insulin in my first video. I will give you the link in the description. Please go through it. Thank you so much for watching. Thanks a lot.